Proteins are going to be the third biomolecule that we talk about. They're going to be, the elements that are going to be involved in proteins are going to be C, H, O, and N. And there are actually more monomers than there normally are for the other biomolecules. So a monomer in this case is called an amino acid, and there are 20 different types of amino acids that make proteins. So it's think about it as like the alphabet. The alphabet has letters, 26 letters, and they can be rearranged in certain orders to make words. Well, that's exactly how these amino acids work. Amino acids, there's 20 different types. They can be rearranged to make different types of proteins. So a protein polymer is called a polypeptide. And the bond that is going to hold two amino acids together is called a peptide bond. So when we look at just the monomer, the amino acid name comes from two things. The amine group is going to be this NH2, and the carboxylic acid, or the acid part of amino acid, is going to be this group right here. Okay, the C double bonded to the O and the OH. Then you're also going to have a third piece. You're going to have something called the side chain. Now the side chain is labeled R because that part of the amino acid is going to change depending on what kind of amino acid we're talking about. So you'll have a central carbon with a hydrogen coming off of it, an NH2 coming off of it, a COOH coming off of it, and then this R side chain. And the R side chain is going to be different, 20 different side chains for the 20 different amino acids. So if you look at them, everything that is highlighted in red is going to be the R group. Okay, so they can be as simple as just an H coming off of it, or they could be as complicated as having ring structures, or even this big thing right here. But these are the 20 different ones. So you don't have to memorize their structure, but you will have to know the general structure of an amino acid. This NH2, the COOH, the R group, and the alpha carbon. So when we look at how they're put together, okay, proteins are formed from chains of these amino acids, and they're going to bond amino acid group to amino acid group. So I'm going to have the COOH and the NH2 are going to bond together right here. So this piece and this piece bond here to make this piece. And you're going to have those chains. And remember that all polymers are going to be made from a dehydration synthesis or a condensation reaction, which means I'm going to make a water. So I'm actually going to take an OH from here, an H from here, and I'm going to bond it right here. Okay, now these proteins are going to be the most vast. There are so many of them, and they can do so many things. They're responsible for pretty much structural support, for enzyme function, for immunity, for everything. And so you can have, because there are infinite amount of ways to put the amino acids in order to make different proteins, then you're going to have proteins that span maybe 50 amino acids to anywhere in the thousands range. And they're very complicated. They're going to have some very complex folding regimens that they undergo because these carbons and these nitrogens are going to interact with one another. So the protein shape, this folding mechanism that I'm talking about, is going to be determined by 4 different things. Okay. When you look at the different types of proteins that you can have, okay, you can have enzymes, antibodies, proteins that are just like hemoglobin, and other structural support like keratin, the stuff that makes your hair really thick, and the stuff that makes your hair grow is a protein as well. Okay, you have proteins embedded in your skin. You have enzymes that are in your saliva to break down food. You have enzymes in your body to break down stuff so that they can build up stuff. Those are all considered proteins. And proteins have 4 levels of structure to them, which means that they will fold 4 different times, okay, or in 4 different levels. The first level is called the primary level, secondary level, tertiary level, and quaternary level. And we'll go over these. Okay, the primary structure is the order in which the amino acids are. Okay, that means that if I have 5 different amino acids, that's the primary structure, is what order I put them in. Okay, so it's just order of amino acids. And their shape is going to determine their function. So again, the order of the amino acids, how they're going to fold, and what they're going to do are all related. So the shape determines the function. Structure determines function. Okay, so in secondary structure folding, you can either have a alpha helix type structural folding, or you can have an beta pleated sheets structural folding. So the alpha helix means that the amino acids are going to interact with one another in such a way that they form a spiral, just like this. If they interact in such a way that they form a pleated sheet, like they fan fold back and forth, it's called a beta sheet. If they spiral, it's called an alpha helix. Tertiary structure is talking about their three-dimensional structure. So that means that some parts of it will be beta pleated sheets, like here, and some parts of it are going to be alpha helixes. Okay, and these are going to fold up into some 3D manner. So this long ribbon represents all of the amino acids in that chain. Okay, and then they're going to curve, spiral, or they're going to pleat. And they're all going to curve up and interact with one another to be a three-dimensional structure. And the last one is going to be quaternary structure, and this is where you can have multiples of the tertiaries that interact with one another. So it doesn't have to be just one long chain of amino acids that makes one protein. It could be 4 separate chains of amino acids that fold into primary, secondary, and tertiary, and then they interact with one another to make the quaternary. So for instance, insulin has 2 different forms. Okay, hemoglobin has 4 proteins that are going to interact. So you see you have 1, 2, 3, and 4 are going to interact to be hemoglobin. Now, the vast majority of your body is going to be made out of proteins. Okay, muscle is protein, enzymes are protein, your hair, your skin, your nails, that's all going to have embedded with a lot of protein in it. And so that is why there are so many different ways that we can make different proteins from the amino acid sequences, because they have so many different roles. Okay, enzymes, which I have a video over those that you need to watch, are proteins that are going to be the ones that are responsible for any chemical reactions. Okay, and that means that they're going to break bonds or they're going to make bonds. Antibodies are made out of proteins. These are going to be structures that are going to recognize and detect foreign substances that shouldn't be there that cause infection. This is what you get injected with when you get like some sort of vaccination. They're also just going to be for structure. Some of them don't have any other function besides just being strong. Your hair, okay? The keratin in your hair makes it strong. Your nails are strong. Your muscles are strong. Okay, spider web, silk, all of those things are a very tensile strength, which means that they can bear a lot of weight without breaking. Okay, cytoskeletons are strong. So this is just for structure. And they are also part of the hormones. Now remember, we talked about lipids and that they are actually hormones as well. Well, some proteins can actually become hormones as well in this sense too. So when we look at the cell membrane, okay, remember we also said during lipids that the cell membrane of all cells is made out of a phospholipid bilayer. But tucked away within that phospholipid bilayer are going to be steroid, cholesterols, carbohydrates, and proteins. And what this does is it just adds a layer of structure. It adds a layer of complexity to the cell membrane, because cell membranes are very important. Their structural integrity must be maintained. And so it's very good to have them very strong, because then they will be maintained a lot faster. And lastly, these proteins will function as receptors. So remember we said the cell membrane was the bouncer of your club, which are your cells. It allows stuff in and out. Well, it has to be able to recognize it as being able to be in there. So proteins that sit on the cell membrane are going to recognize stuff that's trying to get in and say either you're on the list and you can come in, or you're not on the list, you can't come in. And the last one is hemoglobin. Hemoglobin is a protein that's found in blood, and it's the reason why we have different blood types. It carries oxygen, which is very important. Okay, so this is just a slide that will tell you different examples about what proteins are found in your body. I suggest you go back and read them on your own time. And when we talked about the protein folding structure, okay, these amino acids are essential. They're order is essential to being able to figure out what their function is. And so they will have all of these levels of folding, which means that they will fold into primary, secondary, tertiary, and quaternary structures. And there are some things that can actually make proteins fall apart. Okay, for instance, this is what for instance, temperature. Okay, high fever will make your proteins fall apart. And this is what we call denaturing. That just means that the protein integrity of all 4 layers actually ends up breaking down because the heat, because of acid, because of strong bases, something is causing it to not be able to bind to itself to make those 4 levels of folding. Okay, and if they lose their shape, they also lose their function. Okay, they must be able to function correctly, which means that they must have their shape. So what does cause a protein to become denatured? Well, strong acids. Okay, that's why you're not supposed to get hydrochloric acid on your tissue. Remember we talked about the cow tongue breaking down in strong acids and strong bases? Well, what's really breaking down in those is the protein, okay, the muscle part. Strong bases. Heat, okay, whenever you have a fever, you're trying to raise the body temperature in order to denature the proteins of whatever infection is occurring. And in the process, because you're raising the temperature of your body, you're also denaturing some of your own proteins, which makes you feel really bad. Okay, that means that some of your enzymes aren't working right, your hemoglobin's not working right, a lot of things aren't working right. And then something like alcohol or acetone, those are organic solvents, which means that they're made out of carbon. They're just very strong solvents that would be able to break down proteins. So proteins that are denatured lose their quaternary, tertiary, and secondary structure, but they still maintain their primary structure, which means that the order of the amino acids never changes. But if it cannot fold into any of those other 3, it cannot work. So if you've ever looked at eggs are high in protein. Okay, that's exactly what it's saying. An active, high in protein egg means that the structure of the protein has been maintained. That means that the folding mechanism has been maintained. But when you cook an egg, it looks different. It looks like kind of white. Well, that white comes from the proteins being denatured. And here's your vocabulary for the proteins section.